So what's the solution? How does it work? Let me give you an overview of that process. And here's the basic approach, is that we start with the DNA sequence. We then run a gene finder to find where the genes are in the sequence. We then run programs to predict the function of those genes or the functions of the proteins that they code for. And we get a list of protein functions. And then we convert that list of protein functions into a quantitative metabolic model. Well, let's look at each of those steps one at a time. Let's look at the genes finding step first. Well, bioinformatics is actually concerned with many other problems besides those that I'm concerned with tonight. But there's a major paradigm that bioinformatics uses across many of these problems, which is if you want to build a program that recognizes some biological entity, X, what you do is you first gather examples of X. If you want to recognize genes, you gather lots of examples of DNA sequences for genes. If you want to recognize DNA regulatory sites, you gather examples of those sites. If you want to recognize signal sequences that tell the cell when to ship a protein across the membrane, you gather lots of examples of proteins with signal sequences, et cetera, et cetera. And it turns out often gathering the examples may be the hardest part. And that's one reason that there are so many databases in the bioinformatics field, is that people are spending time going through the literature, reading scientific papers to gather specific data that they collect into a database and make it available for different kinds of research. So once you've gathered your examples, then you find patterns that these X's, be they genes or promoters or whatever, have in common. And you build a program that recognizes those patterns. And then you evaluate the accuracy of the program. Hopefully you do it on previously unseen X's so that you are not at risk of overtraining, overfitting the data. And there are two approaches to finding these patterns. There's people staring at the examples, and there's getting computers using machine learning techniques to stare at the patterns, analyze the examples, and try to generalize from the examples to find these patterns. And both approaches are used. Sometimes they're used together. And so in general, if you gather examples of bacterial genes, here's what you find they look like. First of all, the genetic code tells you a lot about how to find a gene. Now notice I'm focusing on finding bacterial genes, which have a much simpler structure than genes in higher organisms. But bacterial genes begin with a start codon. And there are only 3 different start codons that are found in the vast majority of bacteria. And bacterial genes end with a stop codon. Again, it happens there are 3 different stop codons that are found most commonly. Now, one of the things that makes the problem hard is that not every start codon, stop codon pair is a real gene. And so what people want to do is to try to find additional features that genes have that distinguish genes from gene coding DNA from non-gene coding DNA. And one thing they found is that upstream, so here I have 2 sequences that you kind of read like in book order. So if we look upstream of the start codon, 8 to 10 bases, we find what's called a Shine-Dalgarno sequence, which is where the ribosome binds to turn the gene into protein. And it turns out these sequences are not conserved across different bacteria. They're typically 6 bases long, but different bacterial species have different sequences here. So part of developing a gene finder for a new bacterial species is to try to discover what this sequence is. And typically people write programs that look upstream of start codons and try to find conserved sequences that are found in many different genes. But even that is not enough to build an accurate gene finder. And another thing people do is to create statistical models of gene coding DNA that distinguish the gene coding DNA from the DNA outside of genes to look at, for example, what base composition or what base transitions are typically found in gene coding DNA. And you put all that together and you can build gene finders in bacteria that have about 90% accuracy. Now let me tell you a bit about the software tools that we've developed. The software tools we developed are called pathway tools. They actually don't do gene finding. They do some of the downstream analysis from gene finding that I'll talk about in a few minutes. But the basic idea is that what our pathway tool software does is it takes as its input an annotated genome. By annotated genome, I mean that somebody's already run a gene finding program on the DNA, and somebody's already run a gene function prediction program on the DNA, and I'll talk about how they work in a moment. So given that input, what our software does is it creates what we call a pathway genome database for that organism that combines the genome information that is its input with new inferred information that the program computes. And once that database has been created, we then have a component of the software called the navigator that is basically an information querying and visualization tool for navigating through the genome pathway information space. And we also have editing tools which people can use to refine and update a pathway genome database, because in bioinformatics there's no predictor that's perfect, and you often want to refine these databases with respect to information in the scientific literature to correct incorrect predictions. And we can also generate metabolic models from pathway genome databases. And so here's our website, which is called biopsych.org, where we now have about 2,000 genomes available through this website. And here's one of our tools. We call it the Genome Overview. And this is the entire E. coli genome in one screen, where each little blip here is one gene. So although genes are not the same size in general, we're drawing each gene the same size in this particular picture to allow them to all fit on the same screen and be visible. We're coloring the genes according to operon organization or sets of genes that are expressed by the cell together. And we have a variety of tools for navigating through the genome. We have a genome browser where you can click on a gene and see a region of the genome. And so here each little rectangle with the pointy head is a single gene. And we're showing promoters around them which guide the expression of genes. And we can zoom in more and more finely to enlarge regions around a given gene. And we see that we see a gene name, and we see the name of the protein that this gene codes for. If we click on that gene, we'll go to a gene information page that what we've done. So here we're looking at an E. coli database. E. coli being, this is a lab strain of E. coli, not a disease-causing strain of E. coli. And E. coli is probably the best studied organism on Earth, free-living organism on Earth. And we spent about 20 years developing this database, and we have a team of curators who basically spend their time reading scientific publications about E. coli and condensing the information into summaries that are written here along with citations to the literature. They both write summaries for people to read, and then they put structured information into different fields of the database. For example, one set of structured information is the chemical reaction that this particular gene codes for, which is shown right here. This is a tRNA charging reaction. We also have a portion of this gene page that shows the protein sequence of the gene product with different areas highlighted to indicate functional regions of interest on the protein. And we can do things like click on this reaction equation, and we'll go to a reaction page that shows this chemical reaction in more detail. We can click on a metabolite, one of the chemical compounds in this reaction, to go to a compound page that shows all the different cellular reactions that this particular compound is involved in E. coli. So here we're kind of navigating through the space of information, and we can click on any of those reactions. Some of these reactions are involved in metabolic pathways. Metabolic pathways are sequences, linked sequences of reactions that the cell tends to use over and over again. And so here's a pathway for the synthesis of the compound homoserine from threonine, begins with threonine and creates isoleucine. So here we're looking at a sequence of linked chemical reactions, each one catalyzed by an enzyme. Okay, but returning to this question of how do we analyze genomes, after we run a gene finder, the next thing that's run is to predict the function, programs to predict the functions of genes. So here's the problem. Given the amino acid sequence of a protein, so it's trivial to go from the DNA sequence to the sequence of the protein. We just translate according to the genetic code. But given the amino acid sequence of a protein, can we predict the function of that protein? So for example, if the protein is an enzyme, what reaction does it catalyze? If it's a transporter, what molecule does it transport across the cell's membrane? Does the protein bind to a region of DNA to turn some genes on or off? These are some possible types of functions that a protein might have. So there are actually multiple approaches to this problem in bioinformatics, but one of the first developed and probably most commonly used approaches is to compare the sequence of a given protein, a protein of interest, to the sequences of all other known proteins. And then find the protein with the most similar sequence and infer that the function of the protein we're analyzing is probably the same as that of P2. That's the basic chain of reasoning that people use. Well, how do we compare the sequence of our protein to the sequences of all other known proteins that have been sequenced in thousands of labs across the entire world? Well, one nice innovation back in the 1970s or so was the creation of a database called GenBank that for a while was hosted at a company called IntelliGenetics down in Mountain View, but it's now hosted at the NIH. The idea was that the GenBank database would collect together all DNA sequences that any lab ever sequenced. That to publish a paper about your DNA sequence, a journal wouldn't publish it until you submitted that sequence to the GenBank database. So that's how they made people do it, because otherwise people actually wouldn't take the time to submit their sequences to the databases. They had to enforce it with that journal publication approach. And there's another database called Uniprot that collects together all the proteins from GenBank as well as other proteins that currently contains about 23 million different protein sequences. And so the way we do this comparison is we run an inexact string matching algorithm that compares a given protein sequence to all the protein sequences in Uniprot. And here's the kind of result that you get when you do that kind of comparison. What we're looking at here is called a sequence alignment. And what I did here was to take the human ATP synthase protein sequence and to compare it against the E. coli genome and see if I found any matches. And I did find a match between the human ATP synthase and the E. coli ATP synthase. ATP synthase is an enzyme that makes the energy storage molecule ATP, critical metabolite. And what we're looking at here is you read this like a book where you think of this as one line. The top line is the human sequence, and the bottom line is the E. coli protein sequence. And the middle line shows us the degree to which those sequences match. And so there's a letter in the middle line where they match exactly. So here the G in the human sequence matches the G in the E. coli sequence, and the D is an exact match. But I and V do not match exactly, so a plus is shown, but isoleucine, the isoleucine amino acid is chemically similar to the valine amino acid, and so a plus is shown to indicate they're chemically similar. Where there's no similarity, we're shown a space. Where there's actually an insertion in one sequence or a deletion in the other, the dashes are shown. But one really interesting thing here is there's a lot of similarity between the E. coli sequence and the human sequence. Why do you think that is? It does catalyze the same reaction, but why would the sequences be similar? We're cousins. We're cousins. E. coli and humans share a common ancestor many millions of years ago. And since that common ancestor, E. coli and humans have been diverging, and the protein sequences have been changing, but there's still enough conserved that we can recognize that they share those proteins share a common ancestor. So again, we infer protein function by finding matches between the sequences of proteins and inferring that their functions are the same. That inference is not always correct. The databases are full of errors, actually. When that inference is not correct, people try to be smart about how similar the protein should be before they'll make that inference, but that problem is not solved.